RNA isolation. So, RNA isolation has been performed to determine the RNA quality and quantity, which are the important factors for ensuring the accuracy for gene expression analysis and other RNA-based downstreaming applications. Introduction. RNA extraction is a method of isolating total RNA from animal yeast or the plant cell. However, there are many methods available for isolation of RNA, such as organic extraction, that is a triazole method, silica membrane based spin column technology, and paramagnetic particle technology. But the commonly used methods is this triazole extraction because it is sensitive and give high yield amount of RNA. So triazole reagent effectively inhibits the RNA activity, thereby maintaining the RNA integrity, and it facilitate the solubility of the cell component by disrupting the cells to extract the RNA. Principle. So the principle has three main steps, which includes the homogenization, the phase suppression, and the precipitation. So first step is the homogenization. Homogenization is being carried out with the help of triazole reagent. Triazole reagent is a ready-to-use reagent and it has three main components: phenol, guanidine isothiocyanate, and isoamyl alcohol. During tissue homogenization or lysing, this reagent maintains the RNA integrity by disrupting cells and dissolving cell components. Next step is the phase suppression. It is done by addition of the chloroform followed by centrifugation that separates the solution into three phases: aqueous phase, interphase, and organic red phase. The aqueous phase will contain RNA, the interphase will contain DNA, and the organic red phase will contain proteins, cellular debris, and lipids, as shown in the figure. Now comes the precipitation and resuspension of RNA. So, once the RNA is been restored from the aqueous phase, it been recovered by precipitation by adding isopropanol followed with ethanol. Once it is done, we get the RNA pellet. This pellet is been redissolved in the nucleus-free water, which can be stored for the further analysis at -20 degrees Celsius. Reagent required: triazole reagent, chloroform, 70% isopropanol, 75% ethanol, 100% ethanol, and EPC. So first is the triazole reagent. So triazole reagent works by maintaining RNA integrity during homogenization. By the same time, it disrupts and breaks down other cells and cell component. It has three basic component: guanidine isocyanate, phenol, and isoamyl alcohol. So first is the guanidine isocyanate. So this component will lyse the cells to extract RNA. It will maintain the integrity of RNA by breaking down the hydrogen bond between the water molecules and weakening the hydrophobic effect on RNA. It also aids the denaturation of RNase enzyme. Second component is phenol. So this phenol will help to dissolve the broken protein and RNA in itself. So the lower organic phase that is formed is mostly due to the phenol. Third component is isoamyl alcohol. It works as an antiforming agent and sharpens the boundary of the aqueous and organic phase. It ensures the deactivation of RNA enzyme. Next reagent is chloroform. So, chloroform helps in the phase separation by separating the three phases: the aqueous, the interphase, and the organic. Isopropanol. So, RNA is insoluble in isopropanol. So, isopropanol is added to precipitate the RNA, which is obtained in the form of pellet after centrifugation. Ethanol. So here ethanol is been used in the two form. It's 75% and 100%. So the main aim of ethanol is to dissolve all the organic impurity present in RNA pellet. DPC water, that is RNase free water, diethyl priocarbonate. So the RNA pellet is been resuspended in DPC water. It is used in handling of RNA to reduce the risk of the RNA being degraded by RNAs. So basically it reacts with the amine hydroxyl or triol group of protein, thereby inactivating the RNAs. Sample preparation. So in sample preparation, first of all we have to dissect the fish and remove the gut tissue. Now weigh the fish gut tissue. So after weighing the tissue, to homogenize the tissue, we add 1 ml of triazole per 100 mg of tissue. Homogenize the tissue with the help of mixture. For phase suppression, we add 0.2 ml of chloroform per 1 ml of triazole, and after that we invert the tubes for 15 seconds. Now centrifuge the microfuge tubes at 12,000 rpm for 15 minutes at 4 degrees Celsius. This centrifugation will help to separate the mixture into three phases: a red organic phase containing the protein, the interphase that contains the DNA, and upper aqueous phase which contains the RNA. Clean the laminal flow with the help of isopropanol to maintain sterile condition. After centrifugation, transfer the transparent upper aqueous phase to a new centrifuge tube using 200 microliter pipette. Usually, up to 700 microliter of colorless phase can be transferred to the tube. Now comes the total RNA precipitation. So here we have to add equal volume of isopropanol 75% as per the aqueous phase used and mix gently by inverting the tube. Now incubate the tube for 2 hours at minus 20 degrees Celsius. After 2 hours of incubation, centrifuge the tube for the 12,000 rpm for 10 minutes at 4 degrees Celsius. Now, to give a RNA wash, remove the supernatant and wash the RNA pellet by adding 300 microliter of 75% ethanol. Mix gently by inverting the sample for few minutes. Centrifuge for 7500 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant and add 300 microliter of chilled 100% of ethanol. Now centrifuge again for 7500 rpm for 10 minutes at 4 degrees Celsius. In a drying, so we have to discard deep supernatant and invert the tube on a clean tissue. Dry the pellet for incubating for 5 minutes. Clean the agarose gel electrophoresis apparatus with isopropanol. To make the gel, we have to first weigh the agarose. We are weighing here 0.75 gram and adding in 50 ml of tea buffer. To make agarose, weigh 0.5 gram of agarose powder and mix it in 50 ml of the tea buffer. Boil the agarose to its boiling point, and after that, once it came down to a room temperature, add 1 microliter of ETPR into agarose. Now pour the agarose into plate and makes the well with the help of comb. Resuspend the pellet in DPC treated water as per the sample amount. If the pellet size is small, we add 10 microliter. If the pellet size is more, then we add 15 microliter of DPC water. Place the paraffin paper to mix dye and the sample. Now pour TBE buffer into each apparatus. Place the well plates into the buffer system. Now take 1 microliter dye and 4 microliter sample, mix it properly and load into the well. Once the gel has been run completely, observe the band under. So the result, so figure A and B show the RNA images of our experiment, and figure C shows the reference, that is an ideal RNA band. So when comparing figure A and figure C, in figure C there is a marker, that is an RNA ladder, which indicate the sides of this ladder, and on the right hand side is an RNA band. So at some length we can see there is a dark band of RNA which indicates the intact RNA band, whereas we can see the shaded band of the RNA which indicate the shear RNA. Thus, by taking the reference of the RNA ladder, we can know the size of the RNA fragment. In figure A, there is an image of the RNA band we obtained from the fish gut tissue, shows in the agarose electrophoresis gel, whereas the B image show the RNA band in electrophoresis camera.